Alright, this is the continuation of the DNA replication lecture. In this part, I would like to talk about how DNA polymerase does its work. It's still basic chemistry. DNA polymerase is still an enzyme, aka it catalyzes the phosphodiester bond formation between a nucleotide and a nucleotide polymer, which is DNA. So, as you can see here in this diagram, here's the template strand, the old strand that the DNA polymerase is using, and here's the new strand that it has recently synthesized. Because this has already been synthesized, it's clearly moving down in this direction, so it's moving 5' to 3'. And the input that the DNA polymerase uses are nucleotide triphosphates. These are not ATP molecules with CTP or GTP molecules. These are D-ATP, D-CTP, D-GTP, and D-TTP molecules because DNA is a deoxy ribonucleic acid, not ribonucleic acid. Remember that ATP, GTP, and so forth that are used for energy in the cell are based on RNA and not DNA. So you can see here that the nucleotide triphosphate comes in, and this question asks you, what is the significance about the phosphates of these molecules that helps with the reaction? Well, just because it's a deoxyribonucleic acid still makes this triphosphate bond very fairly unstable and a release of a lot of energy when this third phosphate is removed. And in fact, because there's only one phosphate between the riboses inside a DNA or RNA molecule, both of these phosphates come off as pyrophosphate, which is then promptly degraded by a very fast enzyme into just single phosphates in organic, further driving the reaction towards polymerization as opposed to depolymerization. So this is the basic principle of DNA polymerase activity. It will synthesize a phosphodiester bond by allowing this OH to attack this phosphate, releasing this pyrophosphate and forming this phosphodiester bond. And this is how DNA polymerase actually looks like. There's lots of different DNA polymerases, but they all have fairly similar structure. They basically look like your right hand. There's the fingers of the RNA polymerase. Right here. They have some DNTP binding sites, which stabilize the location of the DNTP right on top of the DNA strand that is about to be synthesized. Then there's the thumb of the DNA polymerase, and it encloses the whole reaction site when the DNA polymerase is really going and it has locked itself onto the DNA. The thumb helps to make this, to isolate the active site from the water and allow for this reaction to occur a lot more efficiently, contributing to the persistivity of the reaction, or how fast the DNA polymerase can synthesize new DNA. And then the palm here is where the active site actually is. You can see in this picture right here, there's two binding sites for the insertion of the next DNTP nucleotide and the catalysis site here. And then right here, right next to the thumb, is a third active site of the DNA polymerase, because not only does it have 5' to 3' polymerase activity, it also has 3' to 5' exonuclease activity, which is important for DNA polymerase for fixing mistakes, because it can make mistakes. If certain DNTPs are present in way higher concentration than other DNTPs, they will naturally start out-competing other DNTPs for the binding site, for example, or by random statistical luck, an incorrect base pairing between, say, an A and a C can occur. So, however, DNA polymerase has proofreading activity, which is 3' to 5' exonuclease. Exonuclease means you cut a nucleotide out. you remove it. It's the opposite of polymerase. So let's look at this in a little bit more detail. So let's pretend that this is still our parent DNA strand that has been unwound so nicely by the helicase here. And I'm not even going to bother thinking about which one of this is 3' and 5'. Let's start with this strand because it's easier to hold. So the DNA comes in like this, right? And I'm just going to use my pinky here to hold it in place, but the fingers of the DNA polymerase do not have DNA binding activity, the thumb does. So it's helping it somehow here, but the DNA molecule will go here. There's a little bit of a bend. And the nucleotides will come in through here, and the catalytic site, the active sites are on my palm of my hand. And then as the DNA has been synthesized, the DNA polymerase was here first, and it's moving this direction. So this is the new DNA that has been synthesized. I'm going to wind it onto the pipe cleaner so it actually stays in place. And you can see here, this is how the DNA is arranged inside a DNA polymerase. So the whole reaction occurs right here in the palm of the DNA polymerase. And because this process is so ancient, DNA polymerases, both for eukaryotes and prokaryotes, have very similar structure. In fact, this is a picture of a pol-3, which is the DNA polymerase from bacteria, from Escherichia coli, that's used in DNA replication. But let's look at the eukaryotic polymerase. So here you go. This is polymerase delta, right? Pretty sure. And I got this from the PDB database, and this 3IAY designation is the PDB database code for this crystal structure for polymerase delta, which is, so in eukaryotes, this is the polymerase that will make Okazaki fragments on the lagging strand. So let's go into full screen mode here. So you can see the DNA here, the green and the red. And the red is the newly synthesized DNA, and the green is the parent strand. This right here is the catalytic site of the palm. So let me rotate. Oh gosh, I can't. Come on, bud. Okay, and then around this axis. There we go. So this is how the picture is depicting that you just looked at. This is how the picture. No. So you can see that this is the thumb, these are most likely the fingers, and this is the palm. You can see that the catalysis is occurring right here, and let's zoom in. You can see that this blue structure right here is the next it's the next DNTP that's going to be incorporated into the red strand that's being synthesized. These three dots are the magnesium ions that are helping the reaction. And there, remember how all trinucleotides or triphosphate nucleotide triphosphates are associated with magnesium to counteract the partial negative charge of phosphates. You can see that there's the base, there's the ribose or deoxyribose right here, and then the phosphates 1, 2, and 3 right there. So this has been all aligned up by the position of the amino acids of the active site, and right there. And now let's look at how this amino acid, how this active site looks like in a DNA polymerase. So here's this, again, the picture of the structure if you guys want to go look. And here is the summary of the DNA polymerase reaction. As I said already, the polymerase reaction binds the DNA that has been unwound by the helicase. So the template strand, there's only one of the two template strands of the DNA that's been being fed into each individual DNA polymerase. This is the already synthesized part of the new DNA that the DNA polymerase has made. The DNA polymerase will be moving from here to here, from left to right direction. And you can see that how the electron transfer here occurs. It comes from the OH group, deprotonated or OH group of the 3' carbon of the ribose, and results in the formation of a breakage of this double bond, of this single bond between the phosphate and oxygen, the release of pyrophosphate, and the formation of this phosphodiester bond right here. So if you look at the actual process of what the polymerase does, in this case it's polymerase 1 from bacteria, but it works for all the other polymerases. First, an incoming D NTP is inserted into the insertion site, the linkage is catalyzed, and then the polymerase translocates itself onto the next site, moving the proofreading site where the AT is to make sure that the AT base pairing is correct, and then the green insertion site that was over the T here has now moved over to the G, aka the next nucleotide of the template strand. And here I would like to point out that that even though DNA polymerase reaction is similar, or it's nearly identical between different polymerases, there's lots of different polymerases, DNA polymerase inside the cell. So epsilon and delta are the big guys. If you have kids and know Thomas the Engine, it's the Gordons and Spencers of this world. They're really fast and bulky and are meant for specifically DNA replication. And then there's other less precise DNA polymerases, such as the primase or polymerase alpha, or various polymerases that are involved in DNA lesion repair when the DNA is not being replicated, but just fixed in various positions to maintain the integrity of the DNA molecule. But what we have just learned from polymerase 1 applies to all the other polymerases. They all have catalyzed the phosphodiester reaction. They all have this 3' to 5' exonuclease activity, some stronger than others, some are better than others. Some are faster at making the DNA, some are slower. A primase does not need a primer, it will just plunk some DNA on, some RNA on top of a DNA molecule. And the amino acids involved in the DNA polymerase's active site are sometimes they're aspartate and sometimes they're glutamate. So aspartate and glutamate right here are the amino acids of the DNA polymerase active site, and they're responsible for holding these two magnesium ions in place. And it is the magnesium ions that deprotonate the OH group here of the 3' ribose from this previously synthesized DNA strand, which allows for the formation of the phosphodiester bond between this oxygen and the phosphate, and the release of the second and the third phosphates of the DNTP as pyrophosphate. Because the breakage of the phosphate bonds, because the phosphodiester bonds here, are unstable, the breakage of this bond makes this a favorable reaction, driving the DNA polymerization. So you guys can see here that this is the location where all this is occurring. And because Overwhelmingly, all DNA polymerases also have proofreading activity, which is a 3' to 5' exonuclease activity. There's a second site. This time it's the post-insertion site, right? So this time it's this 3' to 5' site, sort of on the side of the palm region. And this region has more constraints than for the nucleotides, and will detect if the base pairing is incorrect, because a base pairing will produce a bulge in the phosphodiester molecule here, bumping it out a little bit more. So the magnesiums here are held a little bit further from where they are in the polymerase side of the DNA polymerase. As a result, only when there's a bump out due to incorrect base pairing, say A has been paired up to a G, will these magnesiums be close enough to now deprotonate this OH group and then break off this whole entire nucleotide, preventing it from forming. Then when the oxynuclease activity occurs, then the DNA polymerase will back up and try again. This does slow it down though. So you guys can see that the distances here are only close enough for the catalysis to occur if there's incorrect base pairing. And otherwise the new synthesized strand will pass by this catalytic area unaffected. Another important thing is to increase processivity. DNA polymerases, especially the fast-moving ones that play an important role in replication, DNA replication, will undergo a conformational change. Here you can see pole 1 in its open form and its enclosed form, where there's a 40-degree shift in the fingers and the thumb, kind of grabbing onto the DNA molecule tighter, allowing for the reactions to proceed faster. And you guys can see here how the exonuclease decision at the 3' to 5' exonuclease site is occurring. You can see that these guys will fit perfectly, the correct base pairs will fit perfectly into the active site, and thus, however, the incorrect base pairs are placed out of whack a little bit, allowing the actual catalytic magnesium to be close enough to catalyze an exonuclease reaction. So